Hello, my name is Joaquin Larico Nina. I will be presenting a symbiotic filamentous gut fungus ameliorates MASH via a secondary metabolite, CERS6 ceramide axis by Zhao et al., published in 2025. I found this to be quite an interesting read, so I'm happy to be able to present it to y'all and hopefully help everybody learn something about this paper and hopefully have understood the paper and to be able to share it appropriately. So let's get started. So what is the CERS6 ceramide axis? That title was quite a lot, and the background is going to be important to understand what we're dealing with and how it relates to the big picture of human health. So first, what is CERS6? CER6 is short for the enzyme called ceramide synthetase 6. Um, this ceramide synthetase 6 produces ceramide sphingolipids. These sphingolipids are pretty important as components for cell membranes, signaling, and most importantly of all, LPS synthesis, lipopolysaccharide synthesis, and you'll see why in a moment. And an excess of ceramide can lead to metabolic disorders. So the CERS6 ceramide axis, in short, is just CER6 making ceramide. So a biological pathway. Um, so yeah, the first part of that was MASH, so an abbreviation. Um, so is an abbreviation for metabolic dysfunction associated steatohepatitis, but it's actually a smaller part of a bigger label, metabolic dysfunction associated fatty liver disease. So 25% of the world adult population actually have metabolic dysfunction associated fatty liver disease, um, or MAFLD for short. And this figure is growing and is becoming a pretty big concern within medicine. Um, it can progress to metabolic dysfunction associated steatohepatitis or MASH, basically lots of fat buildup on the liver. As you can tell, it's pretty self-explanatory as MAFLD saying fatty liver disease. So it's actually kind of like a range of diseases. It can range from simple hepatic steatosis, which is just like a little bit of fat buildup, all the way to MASH, which is like a lot of fat buildup. And that can even go into cirrhosis, cancer, or even liver failure. So all these very obviously not good things. Um, but there are a few treatment options available. But the key word to notice there is few. There's not much to do once this is fully set in. Um, yes. Why is this relevant? Well, as I said before, I said there's only a few treatments. Currently, the only real way to treat it is a thyroid hormone receptor beta selective agonist, mouthful. Um, it's a recently approved FDA drug, very recently, I think early last year. So not a lot of wiggle room if you're not able to get your hands on something like this. So obviously, development of more important and more effective treatments is very necessary to be able to treat as many patients as possible, especially with how much this disease is growing in incidence. The next part that's relevant is specifically from the title gut filamentous fungi. Um, most research is actually only done on yeast when it comes to gut fungi, and specifically on yeast is the pathogenic effects of yeast, so how it harms us. It can actually worsen MASH in some cases, and we'll see that later in the paper. Um, but determining which specific gut filamentous fungi can adapt and colonize our gut microbiome can help us understand the role in human health, and that's really what this paper is trying to accomplish, is kind of pioneering seeing what is out there in terms of presence in the gut microbiome. So the main research questions discussed here were: what specific roles do gut fungi fulfill in human metabolic disease? What type of fungi even grow in the gut microbiome? So just characterizing what's in there. And then which fungi from those fungi are involved in reducing MASH? As I said before, the main focus of research has been yeast, been Candida albicans, those types of fungi. And just broadening our perspective is going to help us understand our microbiome better. Because even as a focus of being yeast, our main focus has in the gut microbiome has always particularly been bacteria. So this is like a niche within a niche. So it's pretty important to flesh out like every corner that we can in order to see all the aspects that can improve our health. So onto the methodology. Quite a lot. Um, yeah, I'll try my best to explain it all. So to start, uh, the authors developed a somewhat novel technique, phi chip based optimized in situ cultivation system for gut fungi. In short, you can say focus G, as it is referred to in the rest of the paper. This allows for cultivation of previously unculturable gut fungi, and paired with ITS amplicon sequencing, so internal transcribe spacer sequencing, um, this lets us identify all those new fungi that we were not able to cultivate before. Um, what internal transcribe spacer sequencing, in short, is using the sequence nucleotide order of rRNA, a specific sequence of rRNA, to differentiate and identify different fungal species. It's in a lot of ways similar to Illumina, where it's rapid, it's cost effective, and it's pretty accurate. It does share a lot of the same weaknesses, but since we're only looking at a small section of RNA for this, then we can kind of overlook those weaknesses. Next, the authors use mice colonization models. They use a lot of mice colonization models when we're colonizing a specific fungus, our mystery fungus that I realize is kind of spoiled in the next slide. Um, and they also use it to colonize a lot of other fungi, but more than anything, yeast. Since we've already discussed that yeast can actually survive in the gut microbiome, this will become relevant as a comparison point later. Next is RNA sequencing to determine like what, if any, genes affect and let fungi live in the gut microbiome. And then next is hematoxylin eosin, H&E, oil red O, and serious red staining is going to be the backbone of all the staining done in this paper. And this is done to just visualize liver tissue and see exactly more broad, more, I guess, physical effects of the different diets, the different treatments, everything that's going to happen later in this paper. We also use Crelox KO, so using the Crelox KO system to knock out specific genes and see what effects they have on the organism, the mice in whole. And then sequential multicolumn chromatography when it comes to identifying the um, specific molecule that actually helps ameliorate MASH, as said in the title. So a brief overview of the results. Spoiler alert, the fungus in question is Fusarium photons. But here we'll go over the main bullet points to keep in mind while we're looking at all these fun graphs. Focus G is a powerful tool to identify and cultivate diverse fungi. So as I said before, they developed Focus G to be able to actually culture a lot of these fungi that have been previously unculturable, and they're able to make a phylogenetic tree out of it. And a lot of this information, it kind of builds upon itself. I found it pretty important to kind of include most of these figures because it just kind of keeps more and more specific based on the data that we got from the past figure and past experiment. So going to the right, we're going to go from left to right, and then the next row below that. Fusarium photons is an adaptable gut fungus. We identify Fusarium photons as the fungus in question. And we'll see what's interesting about F photons in a second. F photons alleviates MASH in mice models, so we're going to give the F photons to mice compare it to a control diet, well, I wouldn't say control, but compare it to yeast that we know can exist neutrally or slightly negatively in the host. And F-photons inhibits intestinal ceramide synthesis. It's kind of self-explanatory. We know that F-photons can colonize. Now we want to know exactly like how it's helping. Secondary metabolites inhibit CERS6. So now specifically, what is it affecting, and like what is it using to affect it? And then last but not least, spoiler alert, FFC1 is a CERS6 inhibitor. So F-photon makes FFC1 to do that. On to figure 1, focus G. So figure 1A is kind of just like a flowchart of the thought process of focus G. So first, we're going to do 5 diffusion chambers, which typically have always been used for bacteria. And they just culture in situ, allow the what we're culturing to absorb nutrients from its natural environment. So it allows us to culture things that would be very difficult to culture otherwise. So we did 5 diffusion chambers with fecal samples from 5 different people, and incubated with fecal suspension and PDB. PDB being potato dextrose broth, kind of as a control. And then from there, we did ITS2 amplicon sequencing, and that reveals that the diffusion chambers have a similar composition of uncultured fecal gut fungi to, I guess, the diffusion chambers. And then from there, we kind of broadened our perspective. We use a dilution plate method to compare to focus G and see how sensitive, how accurate focus G is. And what came out of that is we did 20 more volunteers, and focus G was actually able to isolate 55 species, while dilution plate was only able to isolate 27 species. So shows pretty sensitive. It can isolate basically double what dilution plate methods could. Figure 1B is using all that data. We're doing high throughput fungal isolation, and we're using all that ITS2 amplicon sequencing data to make a phylogenetic tree based on similarity to, I think it was gene bank data. So in short, we have our 3 most prominent, I don't know if you can see my mouse, but on right below the B, the class distribution of all isolates, we can see our 3 most prominent I guess fungal species were Saccharomycetes, Uretiomycetes, and Sordariomycetes. And within those, our most prominent ones were Candida, Fusarium, and Talaromyces. And this is colored, and the way it's colored is that the blue branch, the Saccharomycetes on the right, the color blue represents a similarity score of 94 to 98%. Let me double check that before I lie to everybody. And then the yellow color on the left, the Uretiomycetes, represents a similarity score of 91 to 94%, and the red nodes is a similarity score of less than 91%. And yes, it was GenBank databases of other fungi. So we know Candida's yeast. Fusarium is what we're, spoiler alert, was what we're looking at. And we're going to touch on figures C and D in a bit. But yes, let's go on to the next slide. Evaluation of gut fungi. So now that we know what's in there, we know that Candida and Fusarium are in there. It was actually determined that those 2 were the only 2 that were identified with both dilution plate and the phyt chip method. So those are the 2 the authors want to go ahead with. We want to simulate the environment that these fungi are going to grow in. So they did a test that simulated the temperature and the oxygen levels of the gut microbiome, and a little bit below that. I'll discuss that in a second. Most of the species ended up growing at 37 degrees Celsius. Some of them couldn't grow more at 30, but it was, I wouldn't say not relevant, but in terms of what we're looking at. Most passed, so it didn't really narrow it down all that much. But when it came to the oxygen test, what we tested, um, what the authors tested was different levels of oxygen. So they had a fully aerobic, I guess, sample, a control with 100% oxygen levels, and two more experimental plates where they did to simulate the small intestine and the colon, so 2% oxygen and 0.4% oxygen levels respectively, to determine what grew. And so that was microaerobic and anaerobic. And consistent with other previous reports, yeasts grew. We already knew this. We know it goes in the gut microbiome. But what else also grew was fusarium, um, which is one of those that I told you about earlier. So it was able to grow anaerobically, and from this, they determined that fusarium was probably the most likely to be able to grow in the gut microbiome. So now, fusarium photons. There was 4 species of fusarium that actually grew, but we're going to look at specifically photons because photons is actually the most present. Fusarium is globally present all over the world, but the most present of those is uh, fusarium photons. So what they did to determine whether photons can actually colonize in vivo in an actual model was they just gavaged or force-fed germ-free mice that had nothing else in their microbiome, or I guess the lack of microbiome, to see if they could detect it in their feces, and they could. They could detect it within 2 weeks. And they also did qPCR to see whether it was present in any other tissue besides the colon or besides the intestine, and it shows it's actually only just in the colon. Very little in the in the intestine, and absolutely nothing anywhere else. Sorry. um, F photons expressed different genes in different aerobic conditions. So I kind of phrased that a little weirdly, but basically, when it's in anaerobic conditions, it expresses more 2 specific genes, FF4063 and FF6139, and they allow F photons to actually grow anaerobically. And in a knockout experiment, they knocked out these 2 genes, and it actually removed the ability of F photons to grow anaerobically. And when blasting these 2 genes, they found no close homologs in any other fungal genome. Um, so we saw before the phylogenetic tree how it was less than 91% similar. This probably will fall into that and would explain why this, of all those fungi, is the only one really that could manage to survive. So now I've said figure 1C and D. Here we are back again. So we can see D within the 2 weeks in germ-free mice, and we also did it in specific pathogen-free mice, so free of photons, obviously, that they were actually present within 2 weeks. And then for figure D, the actual abundance, where it was located. So we see in the cecal contents and colonic contents is where it's most present, but not really any in the intestines, very little, and then absolutely nothing anywhere else. So very limited to the colon in both the germ-free mice and the specific pathogen-free mice. Which I'll say SPF now to to not have a mouthful every time I say it. So now we know what's in people. We know what can actually colonize. We can grow, we can colonize, and now we're going to see what it actually can do. So in mice, back again to mice models, photons can reduce MASH. So the way the authors determined this was they did specific pathogen-free mice. I just promised I would say that quicker. SPF mice versus control mice, and each mice was either fed chow or a choline-deficient amino acid high-fat diet, which I'll just say high-fat diet for short. And then after 6 to 8 weeks, they were given either F photons or C albicans yeast uh, after they've already shown a MASH phenotype. So they're experiencing all the fatty liver disease symptoms, and yeah. So what happened was specifically in the F photons mice, there were lowered levels of basically every liver test you could think of. ALT, AST, hepatic triglycerides, hepatic total cholesterol. We know it's a fatty liver disease, so obviously triglycerides and cholesterols, fats, they went down. And ALT and AST, which are alanine transaminase and aspartate aminotransferase, respectively, are enzymes that are released when the liver is damaged. So they went down too, which means the liver was healing or had time to recover. If we're talking about like physical injuries, lacerations, things like that, they had lessened hepatic steatosis, hepatocyte ballooning, lobular inflammation, and fibrosis. So just less injury to the liver in general. And they repeated this with germ-free mice to eliminate any effects that there could have been for fungal bacterial symbiosis. So the specific pathogen-free mice was lacking just F photons. Germ-free mice is lacking everything. So there's nothing to interact with that could have been like colonized before. So here is figure 2. It kind of just shows what I've been explaining. Figure A is the I guess like the flow chart. The idea is what they're trying to do here. BCD liver weight has gone down. If you can see, hopefully you can see my mouse. I don't know. I should have checked that before I made this final recording. The high fat diet with photons is statistically significantly lower than the high fat diet by itself, and high fat diet with the albicans, the yeast. And this holds true for the ALT and AST, as I was explaining before. The chow diet looks pretty just standard. It is the control. If we're looking at F and G, we're looking at AMFLD specific activity and fibrosis. We can see that for the high fat diet and photons, it is lower as well. And when we're going here to figure 2E, this is where I was discussing before the H&E oil red O, serious red, different staining techniques to visualize liver tissue. And the chow diet control looks perfectly healthy. And if we're going to the high fat diet, the control looks pretty bad. There's a lot of holes, there's a lot of injury, and same with the yeast, the albicans. But if we're looking at the photons, it looks much healthier and much closer to what the chow control diet looks like. So we can visually see, besides just blood analysis, we can visually look at liver tissue and see it looks healthier. And then H through N is the germ-free mice, and it's a slightly different since they did not do yeast for this. So we did photons with the chow diet as well to have like all to see exactly if it even affects chow diets. So much the same, the chow diet and the chow diet with photons is very similar. So it's kind of expected since photons is supposed to really help MASH, and in healthy mice, it wouldn't really be helping anything if it's already healthy, right? Or keeping it in a healthy state. But if we're looking at high-fat diet and recovering from already having MASH, as these mice already had, then you can see just the high-fat diet by itself compared to it with the photons, it goes down in the same categories: liver weight, plasma ALT, plasma AST, histology, the IMFLD activity, and the fibrosis levels go down, expression. And if we're looking, same results basically for the CDA, the high-fat diet, and the photons for the chow diet control. Photons inhibit ceramide synthesis. So now we know photons actually helps with MASH. What exactly does it help with MASH in? So to determine that it actually helped in inhibited ceramide synthesis, they screened enzyme activity, and they revealed specifically lowered ceramide synthetase activity. This is from a previous paper that they did where they did a giant screening of a bunch of enzyme activities, and they developed like a novel method for it. Very, very complicated paper that I did not understand very much, but that was the gist of it, was that they developed that tool to use here, similar to how they developed Focus G for this paper. Photons actually reduces plasma ceramide concentration, and administering ceramide to mice with photons actually reduces their benefits. So it kind of like it nerfs the MASH, nerfs the photons benefits. So we're going to look at figure 3 now. A, as always, is the experimental scheme to identify what exactly is being inhibited here. And They did the activity-based enzyme screening. They did lipidomics because for fats, and then ceramide supplementation. They gave it ceramide to see what exactly it's interacting with, and they saw that it's actually CERS6. So yes, this very VIP scores. You can see the dilutions of 0 to 16, 1 to 16, 1 to 22, 1 to 20, and yeah, this was probably my least understood part. But what really identifies CER as 6, what I could tell, was figure 3G. Down here, CER as 6, it has the lowest enzyme activity when paired with photons extract. So that's what's really like narrowed it down for them. F photons inhibit ceramide synthesis. So we're still continuing on this. Crelox, they did the Crelox knockout as I explained before. They used it to knock out CERS6 activity in child mice and their parent mice. So these mice, they were fed chow or they were fed a high-fat diet with the photons to see exactly what effects did the knockout have. Does it actually affect it that much? Is it just photons? Is it there's no other uh, microorganism that's helping at all? Um, the KO mice showed improvement of liver health um, and lower expression of MAFLD related genes, but parent mice showed more improvement with fed F photons, which means specifically CERS6 is what's being targeted here. And to even further prove that, they overexpressed CERS6 with a lentivirus and it eliminated the benefits of photons. So we're just really, really narrowing it down. And we can see from figure A as our schematic again, we're looking on E, F, and G, liver weight, ALT, AST. We're looking at photons with the knockout, photons, high-fat diet with CRS6. These are all low, but specifically high-fat diet CRS6 with the children, was way higher because since they had their CRS6 knocked out, they can't be lowered anymore. They're not attacking the photons isn't benefiting anything. It's not attacking CRX6 like it is in the parents, which can lower MASH symptoms. Secondary metabolites inhibit CR6. So we were kind of already discussing that, but they further tested this by extracting. They had F-phone extracts, and they tested it on intestinal organoid culture models, and they showed they could reduce ceramide levels, and specifically identified FFC1 to FFC4 with that chromatography I explained earlier, and inhibited the CERS6 activity. They further went down more specific, and they found actually only FFC1 was detected in fecal samples of specific pathogen-free mice. And yes, and it was also in germ-free mice, but only after infection. Then they actually had a LAC-A knockout, and when they got rid of LAC-A, it actually eliminated the inhibition effects of ceramide. So let's discuss this on the actual figure. So figure 4. If we're looking at A, we can see photons extract the control, photons extract, and then photons with the knockout basically puts it at the control. So knocking out LAC-A is pretty important that it is a specific part that actually lets it be impacted. If we're looking at figure 4C, they're looking at inhibition rates with silica gel columns, so chromatography, and they saw that FF3 was actually the highest inhibited. So they saw specifically in that that is FFC C1, C2, C3, C4, with the most being C1. But C2 and C3 also showed, I would say, promising levels of inhibition. C4, not really. So yeah. So now that we've identified FFC1 and we know that actually it inhibits CRS6, we want to kind of know how, right? It binds directly and non-competitively to CERS6. So there's a different site that it binds to and actually modifies the binding site for CRS6 to make to bind to its substrate to make ceramide. FFC1, when given to mice with the LAC-A knockout, actually overcomes those effects I showed you in figure A of the last slide, and then inhibited CRS6 and ceramide levels in parent Crelox mice. Crelox mice. Sorry for the stuttering. So those parents that actually were able to be modified, or actually, pardon me, the parents that had the CR6 to be affected um, gets affected by the FFC1. But when given to the KO Creolox mice, the children that had the silent CERS6 gene, there's no effect, showing really that FFC1 selectively targets CERS6. And then the mice were also given a different MASH progression diet. So this was Gubra, Amylin, NASH. And actually was able to overcome the effects of MASH in this diet as well. So let's look at that in the figure, following figure 6. A is a schematic as always. If we look specifically at figure E, F, and G, we can see the CERS6 double flocks are the highest, while with FFC1 is lower. The knockout is around the same. The knockout of FFC1 is around the same as well. If we look at N and O, similarly, the control is the highest, FFC1 showing that it is the lowest. Staining, we can visually tell the liver tissue is healthier with FFC1, even with knockout and no knockout. So, discussion. The authors developed Focus G to identify and culture fungi, evaluated gut fungi and adaptability to see what even is able to grow on the microbiome. F. potens is a gut fungus that helps reduce MASH phenotypes. That's what they identified. And it also inhibits ceramide synthesis from CRS6 via its secondary metabolite FFC1. So we were able to see that with all the mice models and just really narrowing it down. So pretty interesting. I guess like I wouldn't say the approach because I feel like that's probably a lot of research papers, but just kind of building off of the previous information really kind of piecewise. So conclusion, F. potens provides protection from MASH by inhibiting CR6 with FFC1. Questions? Can the FI chip be used as an approach to culturing viruses? I know there's a lot of issues culturing a lot of viruses, and that kind of impedes the study of viruses in many ways. I've taken a virology class, had virology units in a virology class, and that was a somewhat major point of discussion. And some of them was, how do we study things that we can't really culture? We can't really even like look at. So since these authors looked at the iChip and the diffusion chambers, and which were originally used for bacteria, and they really modified it for for fungus, it'd be interesting to see if someone would be like if that could be used as well for viruses. It'd be a lot trickier, right? Because viruses have to live in host cells, but maybe specific host cells that they have a tropism for can only survive in situ, for example, or like in a specific environment, and this is the only way to be able to observe that virus without having to like extract it from like a person's like organs or whatever. I don't know. What other fungal metabolites can affect colon health? So we already know that yeast can somewhat affect MASH, and we know that FFC1 lowers. What maybe F. potens or a different fungus that can't really survive but would make a metabolite that could affect colon health in a positive way. It would be interesting to identify and characterize these and see what other ways we can improve colon health. And how could symbiosis between different organisms impact how effective FFC1 is in a microbiome? So part of the rationale for making germ-free mice was to see no other impact, it doesn't interact with anything else. There's a possibility that FFC1 can actually interact with other molecules, and maybe that can diminish or enhance its effect. So that would be interesting to study and interesting to approach. Can FFC2 to 4 also be used to treat MASH? So C4 probably not. As we saw, it was very little. But FFC2 and 3 had like a 50, a little bit more than 50% inhibition. So perhaps they could be a weaker form of FFC1 that can be given to more sensitive patients or something along those lines. And then finally, how should FFC1 treatment be implemented? How would it even be given? Like I guess the rationale for how to turn it into a medicine, or maybe have people be given like fecal transplants specifically with F photons to improve FFC1 production and help reduce MASH symptoms. Here are my references. Thank you.